วัสดีค่ะ
สวัสดีค่ะ
ครับสวัสดีครับ Okay, I just sent the PowerPoint to the line, so you can download it and then maybe look at it when we talk about today's topic. We're going to start from. Let me share the screen. Okay, we're going to start from the assignment yesterday about the exploration of the sequence database. So, who wants to go first? Any one of you? Do you want to share the screen? You can make the host, so you can share the screen. Okay. So who wants to go first? First volunteer. Anyone? Anyone? Nobody? Who's brave enough?
So I will go first.
Okay.
My name is Nachman Pat and I searching for.
Do you want to share the screen? I can. Oh no. Okay.
I choose the EMBL or the European Molecular Biology Lab for my database. Actually, the database for bioinformatics of this EMBL is EBI or European Bioinformatics Institute, and it's a part of EMBL, and they also receive the fund from the government of the EMBL member state. For the how often it is updated, I'm not sure. I'm not find the information about it. From the data source, I found that they get it from Genome Sequencing Center, the individual scientists and patent office. For the type of data, there are RNA, DNA, gene expression, protein structure, chemical biology, and ontology. For format file, I'm not sure because it's like have a lot of database inside. So I choose one of website that in this database it has a file format that which is a HTML, XML, FASTA, and FAT file. And for the service they give is they have a bioinformatic tool, data resource, and also have a research public about the protein or gene inside, and also have information about training and education, and also industry program service.
Okay. Okay, so I'm actually quite familiar with EMB, the EBI, because I use it a lot in the past, and not long ago I was using it too. So yeah, as you said, there are so many tools and databases in the EBI services. So actually, I believe that if you look at the database, they actually, and the database actually sync with the NCBI and also the database in Japan as well. So I think it's like they synced the database like every week or something, like once a week. So you can find pretty much the same thing in the NCBI database, the GenBank database, NCBI database, and in the EBI database. So, and you can see from here that this is the tool and data resources. You can find many tools that you can use to analyze the sequence. Like for example, you can do BLAST here for a protein nucleotide. They also have a BLAST program for you. And you can use something like Hammer and Cluster Omega. It's a tool for multiple sequence alignment, and we're going to talk about that next time when we study phylogenetics, like how we're going to align multiple sequence at the same time. And there are also some other databases, like the Ensemble is like one of the most complete genome databases and the gene annotation. So if you would like to study like non-model organism or some organisms, and you also want to know where the genes are and what they are, you can come here and look at the Ensemble database. The UniProt is pretty much like a protein database. So if you want to study protein, you can go to UniProt and search. The PDB is, I think it's the database for 3D structure of the protein. And the expression atlas, I believe is from mostly from microarray and the next-gen sequencing data, the gene expression data. And the Shambo is like chemical database, the database of the chemical compound, bioactive compounds, for example. So there's so many things that you can do on the EBI. They even have training. This is the one that I think you can check it out. If you have time, they even have like the training about how to use their own tools and databases in research. So you can see there's so many things that you can study by yourself. So I would suggest you spend some time with it if you really interested in doing something. Like for example, you can, if you want to study like metagenomics, for example, you can sign up for the course. So it's really huge collaborative project. You know, the EBI is, you know, I don't know how many countries actually participate in this institute, but I think it's a very big one, and there's so many things that quite useful for research. Okay, the next one, please. Thank you so much for Natamon. We'll move on to the next one. Anyone?
Hello.
Hi.
Okay, sorry, I was disconnected. Okay, so when were you guys last time that I went off? So I was talking about the EBI that actually. Okay. So what I said before I left?
Okay.
What was I talking about before I left? So I can continue from that.
I said about the source of data or of the gen bank.
Oh wow, that's it. Okay.
Hello?
Okay. Yeah. So just to continue from that, I was talking about the training resources that you may want to check out on the EBI website. There's lots of, you know, training courses, virtual courses that you can take. I believe for free. You don't have to pay anything, and it's quite useful if you want to learn more about bioinformatics. I mean, there's so many topics that are actually taught by people who really know what they are doing. So I would encourage you to come and check out if you want to learn more. And the EBI actually very big. It's a big institution, and they have so many things that, and some of them we're gonna use. Some of the tools we're gonna use in this class as well. So you're gonna be you're gonna get more familiar with the EBI as we go on. Okay, so let's move to the next presentation. So who's ready? Who's ready for the presentation? Okay, go ahead.
Me? Okay.
Someone one. Okay, go ahead. So which one did you look at?
Gen bank.
Okay.
Okay, I will start with who is maintain the database. The GenBank is built and distributed by the NCBI, which one part of NIH of America. And about the data in the GenBank, they have exchange daily with the DNA Data Bank of Japan or DDBJ and the European Molecular Biology Laboratory Nucleotide Sequence Database or EMBL. So the daily upload from the NCBI server incorporate the most recently available sequence data from all sources. And about the source of data, GenBank they accept a primary sequence data which directly determined by the submitter or the bulk of the submissions. For example, EST and GSS, and the author high put and author high throughput data from a sequencing center. And about the type of data that are stored in the GenBank, most they are nucleotide sequence. For example, mRNA sequence prokaryote sequence, viral sequence, cloning vector, or the phylogenetic. And about the sequence that they have in the GenBank database, there are around 200 billion reported sequence. And about the format that are supported by the data, GenBank they have many file formats to support the data bank, include GenBank, FASTA, graphic, or ASN.1. So, and about of the web service, GenBank they have the e-utility web service or SOAP as a service that support the database.
Okay. So the GenBank is the one that you may actually use a lot, and it's pretty well known for biologists. It's something that people will go to when they want to analyze the sequence data, especially the gene data in GenBank. And as you said, there's so many other things, not only genes, like EST sequence and some other sequences. So it's a huge database for all kind of DNA and RNA sequences. So you can, and also the genome sequence. And here's just the updated statistic that you can go and look at on the GenBank website. You click at the statistic link and you're gonna come to this one. You see here, that's the number of the whole genome sequence, right? 2 million. As of 2017, the number of whole genome sequence was 2 million sequences. And number of genes are 200,000. That's that's a lot. And let's look at this one. Oh, this isn't the number of bases. Sorry. So this is like I'm not sure. It's not supposed to be 2 million. It's supposed to be like 2 billion, right? I can't even read it. So the scale is like, this is 1 million, 1,000. So 100 billion bases, not 2 million, sorry. Because it's the number of bases, but this one is going to be the number of sequences. So a number of sequences smaller, but it's still in like 100 million sequences of the whole genome. So that's a number of in the GenBank as of 2017, which is many years ago. Okay, they actually have a number here you can look at. And it's going to keep growing and growing. So there are so many things that you can do with all this data. And actually you can go to when you want to download, you can download actually the whole GenBank database on your computer if you have enough disk space to store the data. You can go to a GenBank FTP server to download everything. And here's all the files that you can download. And if you use the e-utility that your friend said, you can download it more easily using like the automation tool. You can search to download only some of the sequences or pretty much all of them. Yeah, so everything is freely available for anyone that would like to build the NCBI database on their own server and their own computers for the research. Because sometimes when you want to analyze the data, like search for sequences, like many sequences, like I used to search for like 200 200,000 sequences. I can't do that on the website because 200,000 sequences is going to take me so long if I do it one by one. So I have to use a tool and analyze the sequence, they search for the sequence on my own computer. So I can just download the sequences that I want and then build a database on my computer and then use that to search for the sequences that I want. Okay, and there are so many tools that you can use to search for sequences in a GenBank database, and one of them is BLAST, which is one of the most commonly used tools in bioinformatics, and we're going to learn how to use it today as well. So there are so many other, and the GenBank, okay, this one is showing you the sequences that are stored inside the database, and you actually mentioned the EST and the GSS. So Tamon, do you know what it is? The EST and the GSS.
It's like the another like I'm not sure what they call. It's like the group of submissions that we can send the sequence through this path, and it will share this the sequencing that we are receive, that this genome is correct or not, or trustable or not.
Actually, the EST is for stand for
Expressed sequence text.
Yeah. So
and
it's actually
the genome sequence.
Yeah. So it's not really a group of people. It's actually the sequence. Yeah. So the EST is from RNA. When you want to study the gene expression, you can sequence in the past, before the invention of the next-gen sequencing machine, the shotgun sequencing machine, they just use the technique to pull the mRNA to sequence the mRNA from the cell, and mostly they're gonna get only the end of the mRNA, not the whole mRNA, but just from the end of the mRNA, because they know that at the end of the mRNA there will be a poly A tail, so they can design the primer that can actually match the poly A tail and then amplifies just the end part of the mRNA. So that's how you get the express sequence tag, and you can do that a lot, and then you can kind of get a good idea of what genes are expressed, how much, and also maybe some kind of isoform if you get it long enough sequence from a gene. Or sometimes the gene have different like endpoint, like different isoform have different endpoints. So when you compare mRNA from the same gene, you're going to see that that the gene has more than one isoform. So it's pretty useful, but nowadays people just do next-gen sequencing. Just use the next-gen sequencing instead to sequence the mRNA, because they can get the whole sequence compared to the EST. So actually the DBEST and DBGSS is the database that BLAST can search for. Yeah, but all the sequences that, all the gene sequences that submitted to the NCBI would be reviewed by, by scientists, by curators that are responsible for reviewing the sequences. That's why, and some of them, some of the genes that are highly studied, like well studied, well characterized, will become the reference sequence of the gene. So if you look at the GenBank and you see the reference sequence, you know that those genes are actually quite well known for scientists in the field, and it's more reliable than the genes that not the reference sequence. Okay, thank you so much. Any question? Any question about the gene bank? We're going to come back and use this one again. So you're going to know more about the gene bank and the database later. So, okay, let's move on to another one, another presenter.
Can I present?
Okay, go ahead.
Yeah, for my for my database that I choose is the PIR, the protein.
What is it?
Protein information resource.
Okay.
They are the database that under is maintained by the Georgetown University, and they have many funding that give the fund to this, and especially the in the National Institute of Health or in medical science or something like that. And for the update, they have the update bi-weekly, and the source of the type of data that's stored in this database is the protein sequence database, and they have more than 1 million sequence of protein in this database, and it can use for see the sequence protein and compare the protein. They have the compare the protein to alignment about the similarity or difference between the protein, and the file format that they have XML, NREF, and FASTA format.
Okay, that's all. Okay. So this is my first time coming to this website. I have no idea about the database, but so this one is the database that maintained by maybe a single institution. So I'm not sure what is the. I'm pretty sure they actually incorporate a lot of like data from some other sources, right? But I don't know. Do you know what is the main purpose of this database?
Main purpose, in information that I found, the main of this database is the protein sequence.
Okay, so I mean, there are so many other protein databases. So I just want to know, like, what is the unique or the main the unique purpose of this database? So maybe we have to look into that further. But yeah, it seems like a good, a comprehensive database that integrate data from so many sources. Like, in this picture, you can see they actually, the iPro class integrated protein knowledge base. This is the knowledge. I think this is quite interesting. So they don't just store the protein sequences. I mean, this is my guess. I think they don't understand they don't just store the protein sequences, but they store all kind of information. See, Like they integrate the protein sequences from GenBank, from UniProt database. They also store the gene information from EMBL, from some other databases. And they also store the gene expression. They also store diseases, disease information from HapMap, OMIM, and they also store some literature, like the relationship of the protein, character of the protein from the literature as well, from PubMed. So you can see that they actually collect all kinds of data about the protein. So if you want to know more about a single protein, you can definitely look at this one. Say, let me try. Maybe one. Look at the sample report. So here's a sample report from the website, and you can see that there's a whole lot of information. You can see the reference sequence, in other databases. So if you want to know more about the sequence of this protein, you can search. You can just click the link and you go to the reference sequence in the GenBank database or the UniProt. And you can see the taxonomy of the protein, where it's found. It's actually the human protein. And the name of the protein, which is P53. And the keyword, this gene is associated with so many things, alternative splicing, apoptosis, acetylation, for example. So anyone here knows about P53? Have you heard of it before? What does it do? No idea. Yeah, the P53 actually associated with cancer. So if something goes wrong, like if something happened to the P53, you may have a cancer, basically. And you can see there are so many diseases, like so many kind of cancers that are actually associated with this protein, right? So many you can look at like different types of cancers here. And the ontology is basically a control vocabulary that explain pretty much like the function of the genes, function of the protein, and there's a whole big topic about the gene ontology. So we're not going to go into the detail, but mostly when you look at the gene ontology, you're going to know like what it does in terms of molecular function, what it does in terms of biological process, and what it does in terms of the cell component. So there usually are three main categories for the gene ontology. Like this one is actually associated with, in molecular function, this gene serves as the ATP binding, as a chaperone binding, for example. And for biological process, it actually serves as a serve in autophagy and B cell lineage commitment, for example. And there's also some other pathways from cake databases. Cake is, I think it's a Kyoto something database. It's actually had a lot of information about the gene network, and you're going to learn about that on Friday. And the cake is one of the biggest gene network database. And also the Reactome is also the gene network database. So they actually collect all sorts of data for you here, just a single gene. And so if you would like to know about the information about about a protein, you can come here and looking at everything. So this should be a good starting point. If you want to know about a protein, you can get pretty much all kind of information from here. So this is kind of cool. If you work on a protein, you may use it in your research. Okay, thank you so much, and we're gonna move on to the next one.
Maybe me.
Okay.
My database is DNA Data Bank of Japan, or DDBJ.
Sorry. What's the name again?
DNA Data Bank of Japan, or DDBJ.
Data Bank. Okay, DDBJ.
Okay, I will start. DNA Data Bank of Japan, or DDBJ, is the website database that sharing and analysis service for data from the science research or advanced science, which maintain the database by the National Institute of Genetics, or NIC, in Japan. How often it is updated? I don't have the record of this website, but I see at the release update in last year of this website. I would say that they update that daily or weekly. And the source of the data is came from the primary sequence data, because of this website database is collaboration with the EMBL and GenBank. So they're sharing the database and also the third party data, because they allow to have the third party that can submit the data into the database. And how many types of data are stored in the database? In this website, they have a lot of database, a lot of type of database, such as the annotate or assemble sequence, or sequencing and alignment data from the next generation sequencing platform, or the functional genomic data. And the latest update of how many sequences are there in this database is 2.4 billion sequence in today. Yes. And what file format are supported by this database? I found a lot of file format, but I don't understand at all. So I would give an example such as a fat file format or FASTA, or they say a FASTA like. I don't know what it is. And the service that they provide the management and operation of this database website, and also they provide a supercomputer system and provide a service to search and analyze the biological data, of course. And the last one is the training course and publication. That's all from the DDBJ of mine.
Okay, so this is I haven't seen this database before, so this is my first time as well looking at this one, and I think it's pretty much, I mean, it's pretty comprehensive, and it's already synchronized with the one in with the NCBI database GenBank. So the number of sequences is quite a lot, and you say that they also allow people to submit the sequence to the database as well. Okay, so this is more about the DNA sequence and RNA sequence, right? It's not the protein database, but the DNA sequence, RNA sequence. And they also have the training courses. Quite interesting.
And because of they sharing the database with the GenBank and EMBL. So I think the last time that someone want to present the 2.4 billion, I recognize that we have the same number of sequence in the database.
Okay. So this one is probably used by a lot of Japanese, I think so. I guess because I haven't heard this one before, but I'm pretty sure that they actually mainly used by the Japanese. Okay, thank you. Let's move on to the next one.
No, can I share the screen?
Okay, sure.
I cannot share.
Yeah, you can do it now.
Okay. Share the screen. Home. Everyone see my screen, right?
Okay. Yeah.
Okay. I choose the sweet pot database, and the sweet pot database is about the protein sequence database, but now they are in the part of the UniProt, and now UniProt there are many parts, and the sweet plot would be the uniport KB part. This one, see the cursor, right? And this database will provide that. I will show you. Okay. This one is about the protein sequence database, and the format file is, there are many. You can get at a text, XML, RDF, or GFF, and normally you can get these ones as a photo file or the protein sequence. And for the what are the sort of database, mostly they are come from the protein sequencing นะคะไม่ใช่อันนี้ they're from this one, the obtained by direct protein sequencing, and anyone, and I'm not sure how often they are updated, but they have a part that you can, when you get the information, and you can put the information that you get into this part and submit to them. And I don't know when they are updated, because they need to check first it's correct or not, right? So the next one is what, how many types of data are stored in this database. So there are collect only the protein sequence database, but in the Uniport there are many parts, and each of part they are linked together, and Uniport they also link with the NCBI also. And for the how many sequences are there in this database, they have the okay, and statistic. I found, but now cannot find แล้ว They have this statistic อ่ะsorry นะคะอาจารย์หนูคือหนูกดแท็บเข้าไปไม่ได้เพราะว่ามันบังอยู่โอเคมาแล้ว For the statistic is there are many statistics that they done, but mostly they are about the engagement of this website, but there are separate many types of the protein sequence. And as you can see, they are collect like a chain, peptide, propeptide, signal peptide, transit. There are so many, but they don't have like a total number of their sequence. I cannot see. Maybe they have, but I cannot find. And for the fundings, there are many fundings. But the main one is the Uniport Consortium, which consists of the several European bioinformatics organizations and foundation from the Washington DC, United States.
Okay, interesting. So this is the one, I think it's one of the biggest protein database in the world, and many other databases actually incorporate data from the Uniprot. And I hope you're going to get back to the Uniprot again when we study phylogenetics and the protein sequences for sure. So the last presenter, are you ready? The last one, right? Okay. Yeah. Okay, go ahead.
My topic is the super family database, which is the protein sequence database. For the who maintain the database? This database is come from the University of Bristol. The who maintain is the researcher who is Julian Gosch and Silas Cortier, and there are no data about the how often is updated, but the last version of super family is updated on 218, and the source of the data come from many publications that the researcher collect, and the type of the data that stored in the database mostly is the protein family and genome annotation, and there are also the alignment and the hidden Markov model, and they said that there are 3,200 completely sequenced genome against the hidden Markov model in this database, and the file format that support in this database is the FASTA format, and the service that provide in this database there are many features in this database, such as the sequence search, the domain assignment, the comparative genomic tool, the genome statistics, the gene ontology, the phenotype ontology, and the superfamily annotation, the functional annotation, and the phylogenetic tree. Yeah.
Okay, so what's the name of the database again?
The superfamily.
The super family. Let me show.
There are have to. I found that there have to.
Is that the name of the database? Super family database.
.75 superfamily.
Okay, subfam.org. Okay.
Yeah.
Oh. Oh, I see. So this is quite this is more like a specialized database, right? It's a specific database that show that collect all is a super family, the information about a super family, which is the protein family.
This is an old version of this.
The old version. Okay, let me look at the latest version. Where is it?
Sorry,นะคะ.
This one?
subfam.org ค่ะ
Oh.
subfam. Oh.
It looks pretty much the same.
But in my, but that I found that it's not like that.
Okay, no problem. We can just looking at for some information. So yeah, this is the protein super families, and I'm not sure. I mean, I haven't done any work on the protein super family before, so I don't know what they look for in the protein databases. Anyway, but the hidden Markov models is actually the term that you mentioned about. Actually, it's more like an algorithm that you can use to. It's a model, the machine learning model that built from the existing data. And then you can use that model to scan or search for the protein family inside the protein sequence or the genome. So this is why this is how they actually annotate the protein family in the genome sequence. Because, you know, if you just wait for the scientists to discover all the protein family, it's going to take a long time. So they basically build a model. They use the computer to learn how the protein family look like, and then use the model to scan from the genome sequence. You see, they actually they use a hidden Markov model to scan from 2,400 sequence genome. That's quite a lot. So some of the protein family in this database will be pretty much predicted family, a prediction, not the real. It's actually predicted by the hidden Markov models. But it's still useful, most of the time, the predicted genes and predicted like family like this is actually quite useful, especially when you don't really have any other way to search. But you can test it. You can test them in your lab. So so sometimes it's quite useful to have this kind of predictive models. And there are so many tools actually you can use to scan for the protein coding sequence, exon, genes, and like the non-coding RNA, for example. So it's quite useful, but all the tools that you can use to predict all these sort of things has to be trained or be taught by the real data. So it depends on how much data you have to train the algorithm. And we can talk about that later when we come to that topic. Okay. So thank you so much for all of you, and I think we learned something about the databases, right? We know that there are so many different other databases. Some of them actually kind of integrate into each other. Some of them synchronize with each other. Some of them are unique, more specialized databases. But actually, one of the characteristics that you can learn from our presentation is that the database usually they don't store only the sequence, right? They store some other stuff as well, like some other information about the sequence, about the gene, about the protein. So nowadays it's pretty common for the database to store some other related information. So it's very useful for scientists, right? You go to the database and you don't have to go somewhere else. You can get pretty much all kind of information from one place. And that actually makes the database more useful, and it actually increases the number of people who actually come to visit the database, and maybe increase a chance for them to get more funding. So I have a question for you guys, just to think about as exercise here. Why do you think there are so many there's so many databases that actually store the same thing like DNA, RNA, protein, but in different countries? And why Thailand doesn't have any databases like Japan, for example? What's the reason that Japan has to have its own database? European countries has its own database, and in the US they have its own database. So what's the reason for that? Even though they actually store the same thing, and they even synchronize the database. So they actually store exact same thing, but the database is actually maintained by a different group of people.
May I guess maybe because of about we have many different regions in the world, so we need to have like the centers in the different area like the Europe, USA, and in Asia it's Japan, to collect the data of the livings in this area, to group the sequencings and to connect together and share what how similar with this living in this area and in another area, and share some like in case of COVID-19.
Yeah, but nowadays we have it on the internet, right? So you can submit a sequence, even though you work in Thailand, you have you sequence a gene in Thailand, you can still submit it to the NCBI Gen Bank, for example. So it doesn't matter where you are. You don't need a database in your country, because you can submit it to the NCBI Gen Bank. For example, the SARS-CoV-2 virus sequence also submitted to the NCBI from Japan as well, from China as well. So what do you think is
This is about a lot of data, so they have to have many groups to take care of that data. Can you hear me?
Yeah, I can hear you. So do you agree with your friend? Your friend say because the data is a lot, they need more people to help maintain the data.
I agree with Tangmo, because I think that it's because of in Thailand we have a limitation about the big data maintain the big data, and we need the specialists to maintain this big data, and also I think that we need many of the funding that used in this part. So I agree with Tangmo that we need the specialists to maintain of this big data.
Anyone else?
I don't think this is a big reason of the different database, because of it's based on every database have the same data. So a lot of database is not the major reason that why we need to have many database. I can't think the main reason also, but I guess that Japan would like to have their own database, so they have, but Thailand is no need now. I think.
Me neither. I think it's about the funding. If they have their own data, they can recruit some funding to their own country.
Really? They actually spend a lot of money to maintain a database. Actually, in order to have a good database, you have to spend a lot of money. I mean, a lot of budget from, the tax money to maintain the database. So it costs a lot, but the real reason, the main reason that they all want to have their own database is that it's more about the power in academic, in scientific community. So if you want to be a leader in a scientific research, you have to have your own database. It's pretty much like you want to be, one of the leading country in scientific research. That's why you want to have your own database. Another reason is that what would happen if one day the US just doesn't allow Thai scientists to use the NCBI anymore, for example. That's going to be a big problem for us. So if they control the database, they can control, when they want, like what country they would allow to use their own database, for example. It's their own right? They can close it anytime they want. So if we rely so much on their databases, we're gonna have a big problem one day if those countries they don't want Thailand to use their own database anymore. So that's why the European country, they get together and build their own databases in order to maintain the power, right? So they don't have to rely on the NCBI, which is the US based, and also the Japan. They also want to be a leader in in this kind of research field, so they build their own database. So you can see that the data is very important. It's pretty much like power. So the country that has a lot of data, they will have a much more power compared to like our country, for example. We don't even have I don't know. We may have some like database for rice, for some like some plants that we grow for economy. I believe we do have the database for that, but we don't actually have the database like the EME, the EMBL or the NCBI. One reason is because we have to spend a lot more money in order to maintain the database. So if we don't have that much money, it's quite impossible to do so. But think about those countries. The reason that the US can maintain a very big database, they can spend a lot of money on the database because every year they make a lot of money out of the research. They can sell the innovation from the research. They can sell the product from the research, from the innovation. I remember that actually one year, like a few years ago, the US actually made more than $6 billion from research. So they can make money out of the research. And it's not like our country, we have lots of problems. We can't really translate the result from the research into the industry. Yeah, that's a big problem. So if we can translate the research into product, and we can sell the product like all over the world and make a lot of money, we can invest that back into building the infrastructure and building this source of database. So this is a big problem. So when we want to analyze the data from bioinformatics, we're going to have to rely on data from all these databases. So we should appreciate that. We should be thankful that they let us use for free because we don't have to pay for anything, right? It's a good thing. Okay, and let's go to today's lecture. I can't find my presentation. Okay. So you've heard about different file formats, and definitely one of the question is that why do they have so many file formats? Why they can't just come up with just one format and use like for everything, for example. And when you work in bioinformatics field, you have to be familiar with at least some of them, because some bioinformatics tools will only work with specific file format. For example, if you have a FASTA file that look like this, and you want to use a software that only support the XML, you're going to have to convert the FASTA file to XML and then run that on the software. And you may get the result from the software in another format. And if you want to do something else, you're going to have to convert that format to another format. So just converting, between file formats will take you a while, because if you do it manually, for example, it will take you like a day for it just to convert the file formats, because most of the time you don't only have one file, you may have 100 files that you have to convert, and you have to do that every time that you run the analysis. So it's going to be very tedious. So this is one thing you have to accept, there's no like perfect file format for everything. That's why there are so many formats. And some of the format is very specific to a tool, to a database, but some formats actually pretty common. Like the first one is the FASTA format. This is the format that widely supported in many databases and many tools, as you heard before, right? Like every one of your friends mentioned the FASTA format as a supported format from the databases. So if your tools can understand the FASTA format, you'll be good to go. Like you can just download the FASTA format and use it. So the FASTA format is pretty simple. You have the first line start with the name, start with the greater than sign, and then the name of the sequence in one line, in a single line. Okay, it can be long, but it has to be in one line. So the title has to be in a single line. So basically, don't hit enter. You just write the name until it's complete, but don't hit enter, because if you hit enter, the program will expect that the next line is going to be the genome sequence, the DNA sequence, or RNA sequence, or protein sequence. So this is a simple format. The first line is the title, and the other lines, the following lines, are the sequence itself. And the sequence can be as long as as it is. It doesn't have to. You don't have to put it in the new line like this one. But this one is just it's easier for human to look at the short sequence like this in their own line. But actually, when the computer read the FASTA format, it doesn't care. You can just keep writing the ATTC until the end of the sequence. You don't have to hit enter at all. But this one is a lot more easier for us to look at. And the width of the line of each line, the length of each line doesn't have to be like this long. I think this one is maybe about 60 base pair long, but it doesn't have to be 60. It could be 100, could be 200. Doesn't care. It doesn't matter at all. And this is the very highly completed format, and it's not even for. It's pretty hard for human to understand, but it's pretty much for the machine to read and write. So the XML format is more like it's actually store data in hierarchy, which is in a tree-like format. So you can look at the outer layer and inner layer of the data, something like that. So it's pretty complex data format, and it's not really intended for human to read. But it used to be popular before, like in maybe 10 years ago, 20 years ago. It's very popular. It's a very popular format that communicated, that used to communicate between computers, especially using, like SOAP services, like your friend mentioned earlier. But nowadays, it's already changed. This is not the most popular format anymore. But it's still many old tools still require the data to be in this format to communicate between computers. So it's still available, but maybe in the future it could change to some other format that's easier to understand. And the GFF format actually mostly for the gene annotation. It will tell you the gene name, the type of the sequence, and also the part of the sequence. Like you see here, you can see this gene starting from the nucleotide, the first nucleotide to 2559. And then the first exon is starting from 1 to 357, second exon from 358 to 1200, for example. So you can see this is a gene annotation. So actually, it's going to tell you what part of the sequence is what. Right? There's the coding sequence, exon, and intron, for example. So this is good for the gene annotation. Right? It doesn't store. Some format even store a sequence inside this this file, but mostly you don't see any sequence in here. You have to look at the coordination of the region. For example, you have to look at the first position of the exon and the last position of the exon, and then you have to pull that sequence from the genome sequence, for example. So you don't see the sequence in here. You just know where it starts and where it stops, but not the sequence. And when you study the SNP, for example, like the nucleotide polymorphism, most of the time it's going to come in this format. So if you want to work on SNP analysis, you will see this kind of file format a lot. And this is very specific for GenBank format, and not many tools will support this format. So this is very specific format, and it gives you pretty much all kind of information about the gene, and you can download that. And it's pretty easy for human to read, but if you want to read it using computer, you have to use a tool that provided by the NCBI GenBank. And here is just, you can find this kind of information on the NCBI GenBank, you know, because I believe that every database we have its own way to name things, to identify the sequences. So this one is just the basic naming system of the NCBI GenBank. So for example, you can see the genome has many different categories, right? Different types, different classes. For example, like the one that starts with AC underscore is going to be the complete genome molecule. The NC is going to be the complete genome molecule, and also the reference assembly. So if you want to look at the reference genome, I mean the genome that highly confident or highly has a high fidelity, for example, like it's more reliable, you should look at the NC, the sequence that starts with the NC. And NG will be the incomplete genome region, for example. And the NT is going to be a contig or scaffold, which is pretty much like a fragment of the sequence. It's not a complete sequence from the genome. And NW is going to be a scaffold from the whole genome shotgun sequencing. It's pretty much like the next-gen sequence, the genome from next-gen sequencing, for example. So you can see, you know, even the genome sequence has many different categories. And when you want to use the sequence, you should know a little bit about like what it is. So you know that, okay, if I use the contig, what's going to be a limitation of it? If I use the incomplete genomic region, what's going to be a limitation of the sequence? And if you want a complete, like you want to study something like the whole genome, gene expression, you might want to use the reference genome, for example. So it's you just have to download all the sequences start with NC. So it's good to know, you know, so that, you know, when you would like to work with all the sequences, you know that what you are looking for. Like for example, the NM, which is the mRNA, this one is the protein coding transcripts that usually curated. It means that it's very, it can be trustable, you know, it's more reliable, it should be accurate. I mean, if you use the NM, right? But if you use the XM is going to be predicted protein. So it's not, it may not be real at all. It may not be expressed at all in the organism, but actually predicted by a computer. So if you search for a sequence in the NCBI database and you find the sequences that start with XM, you should keep in mind that those are all predicted model, predicted protein. They may not be real. So if you want to use it in your analysis or your research, you have to keep that in mind because it could be wrong. It couldn't be, it's not real. But, you know, it's quite useful because, I mean, there are so many different animals in the world and there are not enough scientists to actually go and sequence all the genes from our organisms. So the predicted protein coding transcript could give you something, I mean, could give you some insight into what the gene or what the sequence would do or what its function is in other animals, for example. The same thing with the XP. The XP is a protein actually, is a protein sequence actually from predicted model. Okay, you can find this information on the NCBI, and I recommend you to take a look at it, because when you really want to use the sequence analysis in the research, you have to know, so that you know what you can expect. Okay, so today we're gonna continue to the sequence alignment methods that we kind of talked about a little bit yesterday. And today we're going to go into more details about what it is and how it works. Yesterday we talked about the homologous sequences, right? If you remember, I hope that you kind of understand, you can tell now which one is ortholog, which one is paralog. And if you're not sure about that, you can go back and review it a little bit. And it's going to be quite important when you look at the sequence alignment today, because the sequence alignment itself may not be able to tell you which one is ortholog or paralog, or even those sequences are homologous or not. It just, give you the best alignment as much as it can, but you can, it's your job to actually tell if the sequences are homologous or not. So what is the alignment? What is the sequence alignment? So suppose you have two sequences like this one. The first one is THATISASEQUENCE, or you can read it as like that is a sequence. And the second one is this is a sequence. So you can see that you can actually align two sequences of equal length, from one end to the other end, basically from the left end to the right end, one by one. This is what we call the alignment. So when we say we want to do the alignment, this is exactly what we do, right? You align two sequences in a way that the matching characters, the alphabet actually match as much as you can. You can align it in some other ways. You don't have to align them so that the alphabet actually match, but it doesn't make any sense if you do that, right? You can align somewhere else, but it doesn't make any sense, because what we are looking for most of the time when we do the sequence alignment is the similarity between sequences or the differences between sequences. So if you align it in some other way, it doesn't make any sense and it's not useful at all. So most of the time, the biologist will align the sequences so that the alphabet that actually similar or the same should match each other. So you can see from here that the one with the line here, the vertical line, is the one that actually we call a perfect match. It has a perfect match, which is it has the same, it's aligned with the same alphabet. So E aligns with E, C aligns with C. That's the perfect match. And there are two positions, two alphabets here that actually do not align, and we call them a mismatch. So A is mismatched with the I, and T is mismatched with the S. So in this alignment, we have two mismatches, and the rest are matched perfectly. So most of the time, when we look at the similarity between sequences, we're going to try to align the sequences so that the number of mismatches is minimized, is minimal, right? It's lowest. So this one, this is the best we can do, right? You can't do much better than this. There are two mismatches, and that's the best you can do. You have to accept that because the sequences are not the same. But if the sequences are the same, you're going to have like 100% perfect match. So it's going to be a completely matched. So when you compare this, if you want to calculate the sequence, the alignment identity of the two sequences, you can just count the number of mismatches and then calculate it as a percent, and count the number of match, and then calculate as a percentage, and you're gonna get the percent identity, which means how similar these two sequences are. But you can see here that even though these two sequences can match pretty well, like I think it's almost like 90% identical. it doesn't make any sense in terms of biology at all, right? You can't say that these two sequences are homologous because they are not even the genome sequence or the protein sequence. They're just, you know, a sequence of English alphabet. So it doesn't make any sense in terms of biology, but you can still align them. But the problem that we face in bioinformatics is that when we align two DNA sequence or RNA sequences or protein sequences, how do we know that they are actually homologous? Because we can align them in many different ways, but which ways actually make the most sense in terms of biology? That is the main question that we're gonna try to understand today. So sometimes is, you know, in this one, you know, you align two sequences, you know, of the same length. So it's pretty easy. You can align them like from one end to the other end or end to end alignment. But for this one, it's going to be a little bit more difficult because the two sequences are not of the same length, right? This first one is like 7 base long, and the second one is like 6 base long. So this is a problem, right? You can align it in many more different ways, and you can even because of, you know, when in biology there could be a mutation that delete the nucleotide or insert a new nucleotide into the genome sequence. So when you align the DNA or RNA or protein sequences, you are allowed to insert a gap into a sequence in order to align those sequences as well. Like in this one, you see the dash. The red dash here is a gap that you insert into the sequence in order to align. And the gap actually represent the indel, which is the insertion or the deletion of the nucleotide, because we don't know if the nucleotide here is deleted from the mutation or the nucleotide T here actually inserted into this sequence. So we don't know which one is, you know, is the original one. We don't know if this one is actually deleted or this one actually inserted. That's why we call it an indel, right? We don't know, but we need to insert a gap if we would want to align these two sequences. You don't have to insert a gap, but sometimes you need to insert some gaps in order to get the best alignment, right? So this is even more difficult because now you have a lot of choices, like where you're going to insert the gap and how many gaps you want to insert into the sequence. You can insert as many gaps as you want, but it doesn't make any sense sometimes. That's a problem. And you can see in this one, this is the example that I talk about. This is the same sequence that you've seen before, right? The one with this is the different one. The first one has only 5 bases, and the second one has 6 bases. So you see in the table, this is the example table that show you how many ways you can align these two sequences. You can look at the number 1. Okay, if you align with the number 1, you're gonna have one mismatch here, right, and one gap here. But if you align it the other way, you get the number 2. You can have a mismatch here, a gap here, and then you have 2 mismatches here, right? So this one has single mismatch, 1 gap. This way you have 1 gap, 2 mismatches. The third way you have a gap here, and you have, you know, 2 mismatches here. The fourth way you can have no mismatch, but you have to insert pretty much like 10 gaps into the sequence alignment. And, you know, the list could go on like forever because, you know, there's so many different ways that you can insert gap into the alignment. So this is very important when you have when you align sequences that have different length. You're going to run into this kind of problem. Like you have so many ways that you can insert gap into the sequence and align them. But, you know, which one is the best? This is the very important question. Like, okay, so which one is the best? You know, 1, 2, 3, 4, 5, 6.
5.
Okay, why do you think 5 is the best?
Because each letter are matched together.
Yeah, but you have 3 gaps, right? You have to insert 3 gaps into the sequence. Okay, okay. Your answer is 5. Let's keep it like that, and you see what other people think. There's no right or wrong answer here. I can tell you. There's no way you can tell. Okay.
I think 4 is the best because we can synthesize the long sequence and can insert directly to the gap.
Oh, we're not talking about synthesizing anything. We're talking about just aligning them. So the fourth one, I think, is like the worst because it doesn't make any. You don't learn anything from the alignment number 4, right? It's just they don't align at all. Basically, you don't learn anything from that at all. I mean, if you do, if you insert gaps like the number 4 here, you can align any sequence in the world because it's gonna match. Because you can align anything in the world because you can keep adding gaps like this, you know, in the end and the beginning of the sequence. So that's why I say it doesn't make any sense because you can align any sequence in the world this way, but it doesn't give you any information about the homology of the sequences. Any other idea?
I choose sequence on alignment 1.
Because
I think it maybe they have some change in the sequence and it's possible to be the
okay, you pick number 1. So number 5, yeah. Number 4, number 1. Any other choices?
I also think it's number 1.
Okay, number 1.
Because it's have one mismatch and only one gap. It means it has only one mutation and only one indel.
Mm-hmm.
So I think it's maybe the most possible.
Mm-hmm.
I think it's number 5 because there are no mismatch in this and in 5 there are 3 gaps that are lower than the other, like the 6. Yeah.
Okay, so actually this is a tricky question, you know. I can tell you that there's no right or wrong answer here because we don't actually know much about these 2 sequences. And it depends on what you think. It depends on the criteria that you're going to use because if you say that, you know, I care a lot more about the number of mismatch, you're going to choose, you know, different choices with the people who don't care about the mismatch at all, but they care more about the gaps that inserted into the sequence. So for example, if you say, I don't care about the gaps, I only concerned about mismatch. So I will choose number 5 because number 5, you know, you don't have any mismatch, but you can insert like 3 gaps and I have no problem with that. So you can choose number 5, right? But if you say, no, inserting indel is kind of, you know, not likely to happen, especially a single indel, for example. You say that, you know, I would think that mutation, the mismatch, the mutation here is kind of more likely to happen than the insertion because here you have to insert like 3 gaps. But this one, you have only a single gap and, you know, a single point mutation is pretty common, for example. So that's why I say it depends on how you set up the criteria and what kind of criteria you're going to use, and you're going to choose the different way to align 2 sequences. And this is very difficult judgment to make if you don't have a good system. And I can tell you that nowadays we kind of have a system, but it's still not perfect, but it's just good enough, you know, for scientists to use for analyzing the sequences. And you're going to see later that we actually come up with something that it's not perfect, but it's still useful. So maybe in the future, we may come up with some other ways, some other system to evaluate the alignment, and you're going to learn about that in the future if there is one. But nowadays we have some system, but it's not perfect, but they're still useful. So there's so many ways that you can align the sequences. You can do it by hand. You can do some kind of a plot that's called dot plot. I can show you later. And you can use something like the algorithm that's quite complex, like dynamic programming, or which is kind of give you an optimal answer, like the best answer, but it could be slow. Or you can use some like heuristic approach. The heuristic means it's not perfect, but it's good enough. The heuristic approach is something that you can use, and it's going to be fast, but not optimal or not perfect. It might not give you the best answer every time, but it's good enough and fast enough for you to use. So surprisingly, you're going to find later that the BLAST actually doesn't give you the best answer. So every time you use BLAST, keep in mind that BLAST is actually a heuristic method. So it's not perfect. It's not going to give you the best alignment, but it's give you the one that good enough, and it can guarantee you that you can get the result back fast. So this is something that you should keep in mind. And the FastStay actually another program. Actually, it's the name of the program, and the file format comes after the name of the program. So the FastStay actually is a program that works pretty much like BLAST, but it's not as popular as BLAST. So you can use, you can align the sequences by hand. If the sequence is not that long, like this one, only 5-6 nucleotides, or up to like maybe 10 nucleotides, you can do it by hands pretty easily. But once the sequence gets longer and longer, like 50 nucleotides, 100 nucleotides, it's just impossible for you to do by hands. You can use like dot plot or some other methods. And the first way, I would like to introduce you to dot plot, which is a pretty simple and useful tool that you can use to analyze the two sequences, the alignment of the two sequences. And this is a basic idea. You look at you just align the one sequence in the y-axis and the other sequence in the x-axis, or the horizontal way and the vertical axis. And then you look at the alphabet or the nucleotide that match one by one, and you put a dot on that nucleotide, right? For example, you see T and T here, you just put a dot here. You see A here, A here, A here, and A here, you put a dot where it actually matches. The vertical and the horizontal actually match. So this why it's called dot plot, right? It's pretty easy to do. You just keep doing this as dot every nucleotide that matches. But you have to see. You can notice that maybe sometimes a single nucleotide on one sequence can actually match more than one position on the other sequence, like this one, right? A here on the sequence number 1 at this position can match A in other position in many positions on the sequence number 2. And this is pretty normal, and it's actually useful when you're looking at the repeat region on the sequences, because when you look at the dot plot, it's going to show you some pattern that you can tell because of this characteristic. Okay, so this is why I say that the diagonal corresponds to a possible ungapped alignment. So if you want to know how these two sequences are aligned, you can looking at the diagonal. So this one shows you that so many ways that you can align like the sequences. This red line is only a single possible alignment. So there are many different ways that you can align these sequences. So the dot plot can give you just you can see that there are so many ways you can align if you look at all the diagonal line here, but it doesn't give you like which one is the best alignment, but you can visualize how the sequence align. And if you have something like deletion or insertion, you can see the diagonal is going to shift a little bit, right? If you see the diagonal line here and then shift to the right like this, it could tell you that this is something like a gap that actually get inserted. So basically it starts a new diagonal line, right? The first line ends here, and then it starts a new line and go on. This means, but this here where the two line ends and starts, actually a gap that get inserted. That's why the alignment is not connected, because you have to insert a gap into the sequence in order to align. So you can identify a gap here using just looking at the dot plot. Okay, why don't we go to this website and play with this one just a little bit before we take a short break. Go to this website. I think you can type, just type or copy and paste the URL to the browser, and you're gonna see a very cool program that you can use. Okay, so this is example sequences that you can just play with. It's a amino acid sequence. So there are two amino acid sequences that align with the dot plot. And if you look at the top side of the page, you can see that there are some a number here is which is say a window size. The window size basically, instead of you looking at one sequence at a time, like we saw earlier in the PowerPoint, that way we look at each nucleotide at a time. But sometimes you want to look at many nucleotides, like for example 15 nucleotides at a time, and then calculate whether those 15 nucleotides are matched or not, if they're like 100% matched, or they match enough, using some criteria. You just put a dot on that. So if you keep increasing the number of the window size, you're going to see that the dot actually get bigger and bigger. For example, like if you increase the number of window size to 70, you're going to find that each dot represents 70 nucleotide amino acid that actually match according to the scale, according to some criteria that I will going to explain later. So if you want to see how the dot plot looks like for a single amino acid, you can just change the number here to 1. And you're going to see this is how it looks like if you set the window size to 1, which means you're looking at each amino acid one by one. So you can see here it's pretty clear that the diagonal line here is the one that showing you that two sequences are aligned pretty well. So these two sequences are very similar. That's why you see a black line on the diagonal line, right? And you see a lot of noises, somewhere else, because as I said before, one amino acid on sequence 1 could match many like the same amino acid on many positions on the sequence number 2. That's why you see all the noises around the diagonal line. That is pretty normal. So if you want to reduce the noises, you have to increase the number of the window size. See? So when you increase the window size, you're going to see less noises. And the diagonal lines will be more clearer for you to see. So this is how the two sequences align, right? So the diagonal line here. And you can put a click at the point on the graph, and it's going to show you the sequence at the bottom, how these two sequences are aligned. You can just, click on the line, and you can investigate those sequences. Okay. Let me get back to the PowerPoint. So this is example of one sequences that align, and you can see there's a huge pattern at the top. It's more like a rectangular black dots here. These actually represent the repeated region of the sequence. So if you see something like this, it means that there are repeated sequences on the two sequences. So sometimes if you want to know if there are some repeat on your sequence, you can just align the same sequence to to itself, and it's going to show something like this. So this is an example of one of the sequence that has a repeat region in this area. So when you align the sequence to itself, you're going to see a square like this. So that's the sequence, that part of the sequence that have a repeat. And you can try by my small example here by just copying and pasting the sequence on a PowerPoint. Let me show you. Okay, you just copy the sequence here. The sequence that say repeat sequence. This is a very short one, and I just want to show you that if you have a repeat within the sequence like A, G, A, G, A, G, A here, many places, how it's gonna look like on the dot plot. So you just copy the sequence from the PowerPoint and then paste it on the website, either sequence number 1 and sequence number 2. Okay, so I just click sequence number 1 and then delete the sequences here and paste the sequence I copied from the PowerPoint, and then delete the sequence number 2 and paste the same sequence. And then I'm gonna reduce the window size, make it smaller. So this is a single nucleotide, right? Looking at one nucleotide at a time. So if I increase a single, the window size a little bit, yeah, you can see the square in this area is getting kind of more apparent. And those squares are actually the region that has a repeat. Because you see that already on PowerPoint. We have two region that actually repeat region on the sequence. So you're gonna see it like a square like this, the 4 with the 4 corners. That's why I'm showing you that this sequence has repeat in those regions. I just want to show you to know that this is one way that you can align the sequences, but you're not gonna use this kind of tool a lot unless you want to play with or investigate the sequence by yourself. But most of the time, this is not going to be the one that you use in bioinformatics research. But it's good to know that, you know, there's a tool that is available, and it's good to know that you can do this. And I'm going to explain the Blossom 62 later, because you're going to have to know what it is. But for now, I'm just going to move on to the next topic. It's just, the Blossom 62 is just a score ring metric that you can use to score when two sequences align. And in this one, the two sequences mean the two sequences of the size of the window size. Like if you set it to 5, it means that each 5 nucleotide we have to align with some score that, you know, calculated from the Blossom 62 scoring matrix. And if the score is high enough, it's going to put a dot in that window, which is the 5 nucleotide long. So if you increase the window size more and more, you know, you're going to lose, like you're not going to see the repeat, you know, it's going to become, it's going to disappear basically, because the window size is just too big, and it actually cover the part of the repeat. That's why you don't see it. So if you want to study the repeat region on the sequences, you have to, you know, kind of play with the window size a little bit, because sometimes it depends on how big your repeat is. You know, if your repeat is very big, you have to increase the size of the window. If your repeat is small, you can reduce the size of the window. Okay, let's take a 5-minute break, and then we're going to come back to finish the course. We have maybe 1 hour left. I think it should be enough. Okay, let's come back in at maybe 3 o'clock, 3 p.m. Okay. Okay, are you guys back? Okay, let's continue to the next topic. So there are some points that I would like you to remember when you do sequence alignment. The point one is that I say this one before, you know, two sequences can always align. So for example, if you just insert all the gaps like this one. Oh, sorry, I have to share the screen first. Okay, so the first point, you know, two sequences can always align, especially if you insert gaps at the end of the sequences like this. It's going to be just, you know, gap align with the sequences. So you can always align two sequences this way. And you can also align the sequences this way as well, but this is not what normally done because it doesn't make any sense to insert all the gaps between the sequences like this, because you can't tell, like you cannot know how many gaps should be in this area, right? In this one, you insert the gaps, like 4 gaps between G and C, but how can you know there are 4 gaps? There might be 2, 3, or maybe more than 4. So it's just impossible. So this is something that we don't do at all. We don't align gaps, 2 gaps, because it doesn't make any sense, and you know it's hard to tell the number of gaps that you want to insert. Okay. But in this one is okay, you know, you can insert gaps like this, is fine, because this is gaps aligned with the sequences inside inside the sequence. And when we talk about the sequence alignment, especially the pairwise alignment, means you align only 2 sequences. There are some terms that I would like you to know. The first one is the global alignment. We've seen this one before. Basically, we align 2 sequences end to end, like one end to one end. So they have to be of equal length. They have to have the same length. And for the local alignment, you actually don't care much about the entire sequences. You just want to align a region inside the sequence, just one part of the sequence. So for example, this one, one region of this sequence can align to maybe 1 or more regions on the other sequences, and that's fine for local alignment, but not for global alignment. And these 2 ways to align sequences will have their own use. So when you're going to see later, like when you should do global alignment and when you should do local alignment. And also the overlap, you know, you can align sequence to overlap like this, and this one is going to be pretty useful when you want to like assemble or stitch 2 sequences together to form a longer sequence. So if you have 2 sequences and you know the end of the 2 sequences overlap like this, you can actually merge them to get a long sequence. And this is usually done in genome assembly or sequence assembly, because you know when we do assembly, we want to form a longer sequence from like all the small sequences, but they have to be overlap, maybe more than at like the number of the sequences nucleotide that overlap has to be long enough, you know, in order to put them together. Okay, so I say this one already, and let me, sorry, when I use the laser pointer, I cannot control the zoom. So for local alignment, the local alignment is appropriate for finding a specific conserved region. So as we saw yesterday, when I show you the conserved region of the, I think it's like I don't remember what gene, the histone H1 gene. You can see that, you know, not all part of the sequence will be conserved among like species, among genes. So if you align like using the global alignment to align sequence from one end to the other end, you may not see the alignment of the conserved region or the domain of the protein inside the sequence, because when you align from end to end, and especially when the sequence is not of the same length, doesn't have the same number of nucleotide or amino acid, you're going to have to insert the gaps. And when you insert the gaps into the sequences, sometimes it's gonna, it's not going to give you, it's not going to show you the alignment of the domain within the sequence. So just keep this in mind, that if you want to compare or align the sequence, especially the sequence of organisms that related but not closely related in terms of evolution, you would like to use the local alignment. And also when you want to align the protein sequences, you may want to use local alignment in this one. If you care about the domain, the protein domain, or the regions inside the sequence, you should use a local alignment. There's so many algorithms that developed like since 1970. So this is not new. The bioinformatics field is actually not new. It just became highly popular due to the search of the data from biology. But it's been like since like 1970. So it's not a new field. And Middleman and Wunsch actually developed the algorithm that do the optimal global alignment. So it could give you like the best answer for global alignment, but it's going to be slow. And later on, maybe like 11 years later, because in the first in 1970, they actually they started from working on the protein sequences first. People can actually sequence the protein sequence, the amino acid, long before the DNA and RNA. So they can sequence the amino acid, and maybe the sequence is not that long, so they can use the global alignment to study the protein sequence. But later on, when they sequence more and more protein, they would find that some of the proteins actually more important. Some part of the protein actually more important or more conserved than other parts. So the global alignment will not cannot give you that kind of cannot help you find the protein domain inside the sequence. So that's why they developed the local alignment 11 years later, because it takes so long in the past to sequence more and more protein sequence. It's kind of slow process. So it takes so long for scientists to realize that the method that they're using were not effective enough. So they developed a new local alignment. But still, because they compare only maybe pairwise alignment, so they can do the optimal alignment. It could take them a while, and they can wait. But later on, when the number of sequence, getting more and more, it's not practical anymore to do the optimal alignment. Like you can't just looking for the best answer every time, because it's going to take you days or weeks, to get the answer, and it's not going to be useful. So later on, they just develop an algorithm like FASTA or BLAST. They actually use some kind of heuristic approach. The heuristic is basically you just don't look for the best answer anymore. You just provide some rules so that it can speed up the process and get you pretty much somewhat useful answer, but not the best answer. But it's useful enough that nowadays many biologists don't even realize that BLAST will not give you the best answer. They just think that whatever BLAST give you is like the best, but it's not. It's not always the best. It could be the best, but it's not always the best. Okay, so come back to the questions that we discussed earlier. So how can we gonna systematically determine the quality of the alignment, right? We say that we can have many different rules, many different point of view or criteria to use to judge or to determine the quality of the alignment. But how can we do that in a systematic way, not just, arbitrary, just like whatever you like. It's just like in a systemic way. It's a systematic way. So this is very important question that bioinformaticians actually work on for a long time. Right? And you have to remember that, the purpose of doing the sequence alignment is not just finding the best alignment. You want the alignment that actually give you what actually happened in nature, in biology. So even though you get the best alignment, but it doesn't true in biology, that's not useful at all, right? It's not useful either. So you need some kind of alignment that actually may not be the best, but actually real or true in biology, in the real world. So what people thought was to come up with a kind of a scoring system, right? It's pretty much like what you learn in the class. The teacher has to give you scores so that you can tell like who is actually does better than the other. So without the score, it's hard for you to judge or quantify the quality of the alignment. So, but the problem is that like this one, if you say if T match with T or Q with Q, U with U, you give like 5 score, for example. Or if you have a gap, you're gonna, or mismatch, you'll get a -1. This is very simple scoring matrix. But the problem is that, if you have different rule for scoring, you're gonna get a different score. And you can't compare the score between different systems. So that's a problem, right? You have to use, in order to determine the alignment, you have to use the exact same scoring system. And that scoring system will have to make sense, right? If the score is high, it should mean a better quality of the alignment. If the score is low, it should mean the low quality of the alignment. And also, if in terms of biology, if score is high, it should give you a more homologous sequences, not just a random sequences. So this is very difficult, right? You have to come up with a scoring system that reflect the quality of the alignment and also the homogeneity of the sequences so that you can use it in biology. So people actually come up with so many different systems, right? And we're going to talk about them, some of them here. So the first one is they just look at the amino acid substitution preferences. So basically they just collect all the sequences that they can find at that time, and then they look at what is the likelihood, what is the likelihood of the one amino acids that can change to other amino acids. For example, what is the likelihood that A at the same position can change to S, for example. Arginine, convert to serine, for example, and they just look at all the sequences and calculate the likelihood. Like, for example, like out of 100 sequences, they find A that convert to S like 5 times, for example. Cool. So the likelihood would be like 5%. And then they did that, for all the sequences, all the amino acids, and they come up with a score that reflects the likelihood of the mutation that changed, the conversion that changed the amino acid between one amino acid to another amino acid. And some people they build a model from the evolution. So basically this is more like a theoretical approach. So they just, use some advanced mathematics to build a model, the evolution model, and then come up with the matrix. And you have to keep in mind that, when you look at the substitution of the amino acids, most of the time the amino acids that have the same properties, like physical or chemical properties, it might not change the property of the protein that much. So it's going to be more common that amino acid with the same group can convert between each other, but the amino acid with different groups may not like substitute that much. So this is one other thing that people thought about at that time. So they just, you know, give the score, higher score when the amino acid in the same group is substituted by the amino acid in the same group, and give the lower score when the amino acid in the same group is substituted by the amino acid in a different group. For example, the amino acids are actually hydrophilic, you know, and it's not likely to get substituted by the amino acid that is hydrophobic because they're in the opposite property. But it happens, you know, but it doesn't happen a lot. So you would keep the score, lower score for that substitution, and give a higher score for the amino acids that have similar properties. And the empirical data is the one that I said earlier. You know, they just collect all the sequences and calculate the likelihood from that. So there are many different ways, and as I said before, you know, we don't know which one is the best, but we just choose the one that works best for your project and for your research problem, because sometimes it depends on how they collect the data, how they think about the evolution model, something like that, that can make the scoring metrics different. And some of the metrics will work well for your own project, but not for other people's project. So this is a substitution matrix. Basically, they're looking at the likelihood of the amino acid that gets replaced by another amino acid or gets substituted by another amino acid. So there are two of them that are highly popular, and we're going to use two of them today to see what happened. And the BLAST program actually support these two scoring metrics, so you can switch between PAM and BLOSSOM and see what happens. So the first one, the PAM, is called, it stands for Point Accepted Mutation. So this one, they're looking at, basically they collect all the sequences and then calculate the likelihood of the substitution between amino acids. And they collect the sequences from organisms that have different like distance in terms of evolution. So the numbers here mean the distance between the organism. So if you see the PAM 250, it means that they actually collect data from the organisms that quite far apart in terms of evolution. Maybe, for example, like bacteria and human. So these organisms are not close, they're far apart. So if you want to align sequences from the organisms that closely related, like human and mouse, humans and horse, or cows, you may want to use the PAM with a lower number. On the other hand, you know, for BLOSSOM, it's actually looking at the metrics. They use the similarity to calculate the substitution matrix. So the number in BLOSSOM, you know, the higher number is more suitable for organisms that closely related, and the lower number is more suitable for organisms that actually far apart in terms of evolution. So you have to remember one system and then you know the other system, right? If you remember the PAM, that the number here means the distance in evolution. So the higher number, the greater the distance in evolution. But for the BLOSSOM, it's the opposite, right? The higher the number, the closer the organisms, and the lower the number, the farther the organisms in terms of evolution. And there are some other metrics that, you know, built for something else, like intramembrane protein or the hydrophobic transmembrane. So if you work on those proteins and you want to align those proteins, you may want to use these metrics instead of PAM and BLOSSOM, which are just a general purpose protein, general proteins. So for the BLOSSOM matrix, actually they collect a shorter part of the protein, not the entire protein like the PAM matrix, but they collect just small part of the protein and, you know, calculate the substitution rate of each amino acid within the short amino acid sequence. And if you want to know more about the detail, like how they build the matrix, you can go and read, but it's quite complicated. It's not that easy. You know, the way they calculate the score is it's not that easy. You know, it requires a lot of math knowledge to understand. And here's an example of the Blossom 62, right? It's pretty easy to understand when they actually show you the matrix. You know, how they get the matrix is difficult, but once they get the matrix, it's easy to understand. For example, if you see, if you align two sequences, you know, and you see C match with C, you just skip the score 9, for example. And if you see C match with D, you can just keep it -3 because it doesn't happen a lot. You know, it's not likely to happen. The same thing, if you see like E and D, you're going to give it 2. So this means that E and D could happen, but not as often as, you know, maybe E, maybe like not as often as some other amino acid. So this one is Blossom 62. This one is a default that BLAST uses because from what people have analyzed, have done before, they found that if you don't know anything, you just choose this one. You know, the Blossom 62 will work for pretty much everything in general. And if you're not satisfied, you can change the matrix later. But if you don't know what to do, you just start with 62. And this is the default matrix that BLAST set for users. And this is just the example of, you know, the animals that are closely related to one another in terms of evolution. And these are all the animals that are quite far away from human. So you have to, you know, have a good understanding of the tree of life, you know, phylogenetic of the species that when you want to compare the protein sequence or the DNA and RNA sequence, you can choose, you know, the right matrix. Because if you want to search for gene in maybe elephant shark or little skate, for example, and you want to use the human, if you want to find the homolog, for example, the homologous sequences of the human gene in these organisms, in like shark, elephant shark, little skate, you might not find it if you use a matrix that not suitable, like Blossom 62 might not be enough because these animals are far away from human. You have to use maybe Blossom 50, for example, in order to find these species. Okay, this is a very difficult question. What about I give you this as an exercise, you know. We're not going to give you any assignment today. I'm going to give you just this one. So after this class, you know, try to answer, you know, these 4 questions, and then we're going to come back and discuss this tomorrow after the presentation, the paper presentation. The first one is if you want to align sequences from closely related species, what matrix are you going to use? If you want to align sequences from distantly related species, what matrix you're going to use? You can choose both PAM and BAM and Blossom. Okay, just say I'm going to use PAM this, Blossom that. So so you have a good idea of what you're going to use. The first 2 is pretty easy, right? It's pretty obvious. You can answer right away. But the second, the last 2 might be a little bit tricky. You know, the question is, if you want to align short sequences, what matrix are you going to use, and why? And if you want to align long sequences, what matrix are you going to use and why? And this is going to be very important in practice because some of you, like in the morning, earlier today, your friends just show the bacteriocin, the sequence of the bacteriocin, which is quite short, right? It's how long is that? A bacteriocin? Chakan.
39 amino acid.
Yeah, only 39 amino acids. So if you want to align or search for 39 amino acids long, the sequence that match 39 amino acids, that's pretty short, shorter than 100 amino acids. What kind of matrix that you are going to use to get a better alignment? So this is just an exercise, okay? Try to answer these four questions by yourself, and we're going to talk about this one tomorrow. Okay, this is example, when you use different metrics, what happened when you use different metrics. If you use the below sum is basically looking at the similarity. So the higher the number means that two organisms are highly related. So suppose that D and H, are two different organisms. I think maybe H is human, D maybe a fruit fly or something. And if you use the below sum AD, it's gonna match a lot like this, but it's gonna leave out a lot of amino acid at the end, right? Because because of the scoring system, it's gonna assume that the sequences are highly similar. That's why it doesn't care about the rest of the amino acid that doesn't match that well. But if you use the Blossom 62, you're gonna get an extension of the alignment from the Blossom AD, because now, even though you don't have a good match, like a very good similarity, but the Blossom 62 can still pick up the alignment, because you still have P and P here, E and E here, T and T here, and P and P here. But for the AD, it just doesn't care, because the scoring is not good enough, and it's just cut the part that is not similar enough out. So that's why I told you before, is have a good effect, when you use different scoring metrics. So if you want to be strict, like I want to find only the sequences that highly similar to my organism, you just use the Blossom AD. And if you kind of want to be a little bit more relaxed, you can use the Blossom 62, and you can pick up sequences that could be homologous sequences in distantly related organisms. That's why I say, you have to have a good understanding of biology, and you have to have a good understanding of how the program works, and then you can decide how you're going to use bioinformatics for your research. And if you switch to like a little bit more extreme, you go to Blossom 45, you're going to see now it doesn't only find the homeodomain. It finds a lot more than that. Like on the top here is all other parts that actually match, but it's not the homeodomain, right? You get a lot more stuff. This is because the 45 will it's more relaxed, and it's going to give you a lot more match, a lot more amino acids that match, even though they're not highly similar. Okay, so if you just only care about the homeodomain, you can just use the Blossom AD. If you want to look at something else in some other organisms, you can use like Blossom 45. And the problem is that we actually don't have a good rules or a good guideline on how you're going to decide which one to use for your own project. Maybe sometimes you just have to try, Don't go to BLAST and then, just insert the sequence and hit submit and then wait for the result, and that's it. You just have to try. Sometimes you have to try a couple times to see, okay, if I do this, this is a set of the sequences I get. If I do this, this is the sequences I get, and then you can compare the result. So don't just use it as more like an automatic machine that you can just put the sequence in, and then you get the result out and you're done. That's where it's not really a good way to do bioinformatics, because you don't even know if you actually you may miss something, or you may learn something more if you try a couple different like scoring metrics or some other parameters. You can get something else that might be useful. So play with it a little bit, so you don't miss something that might be important. But that's actually what happened to many biologists, because they don't really understand this kind of stuff. So what they do most of the time is just input a sequence, wait for the result, and then they go with the first one that matched, the first few ones that actually matched with the query, and then they think that is the best answer from BLAST, which is not the case. Okay, this is the very important. It's more like a pitfall or gotchas of doing the alignment with gaps. Gaps occur due to either insertion or deletion or indel for short. Insertion of gaps is necessary in sequence alignment, but insertion of too many gaps can lead to meaningless alignment. So most of the time, the algorithm that actually align the sequences will penalize or give them negative score, or deduct the score every time they insert the gap. And a single gap is not usually found. So usually a single gap is not likely to happen. In biology, you see like 2 or 3 or 4 more gaps in the same position, but not one gap. So one gap is mostly a result from the maybe error in sequencing. So if you see a single gap, you should be skeptical. Like this is not really. It could be from most of the time it's from sequencing error, not the real gap. And every scoring matrix will give the score, different score for the gap. That's why this is quite, you have to know that too, right? If you change the score of the gap, insertion or deletion, you're gonna get the new result. And most of the time, we don't really want to play, want to change the score of the gap, because it's quite complicated. It's actually fine-tuned a lot for general purpose. So unless you know what you are doing, you can change. And actually in BLAST, when you use BLAST, you can modify the score of the gap too. You can change it. If you think you can get a better result, you can change it too. And there are some amino acids that should not. It's quite important. They shouldn't be deleted at all, like tryptophan. Like every time a tryptophan gets deleted, it means that they actually it aligns to the gap. It's going to get penalized more, because tryptophan, it could be very important amino acid. It shouldn't get deleted easily, for example. So people have done a lot of research, have done a lot of study, and then they come up with a scoring matrix that they think is the best for us to use. But it's different people will have different systems. That's what happened in bioinformatics, and it's hard. You have to make the decision yourself at the end of the day. And this is the way that people align the sequences. I just show you here so that you know that they don't actually, when we talk about the optimal alignment, they don't actually align the sequences by hand. It's impossible to do that, because there are so many different ways, and it's hard for you to get the best alignment, the alignment with the best score, if you do it by hand. And if the sequence is kind of long, it's going to take you like forever. So this is just the algorithm that people come up with, and you can Google it if you want to know more about it. I mean, there's lots of video on YouTube that can teach you more in detail how you can do sequence alignment using dynamic programming. But for most biologists, you just have to know that it's a trick that you teach. You tell computer to split the alignment into a smaller part, and then you calculate the best score for each part, and then you connect all of them together to get the best score overall. And that's the way it does, but it's quite complicated in terms of computation. But I want to show you a little bit. There's a website that you can go and play to learn more about how the dynamic algorithm works, and you can see a much better picture of how BLAST works as well. So you can go to the website. just copy and paste the URL like before. This one is the Needleman one algorithm, which is the global alignment that can give you the optimal alignment. Just the demo, okay? You don't have to. You can play with them with this one later, but I just want to show you a demo, a small demo, a short demo, so you can see. So, for example, if you have two sequences that you want to align, this one is only 5 nucleotide long, and this is only 4 nucleotide long. How are you going to align these two sequences with the best score based on the similarity? And you can see here, this is a scoring matrix. It's pretty simple, right? You just, align, if you align the nucleotide perfectly, you get 1. If it's a mismatch, you get -1. And if it's a gap, you get -2. This is pretty simple. And you can change it. If you want to try to play with this one, you can change. And it's going to change the result as well. So suppose we use the default value and then we look at the alignment. So this is what happens in dynamic programming. It basically calculate like the best way that you can align the sequences for you. So just ignore this table because it's quite difficult to understand, quite complex to explain for now. But looking at the result here, you see that this one, the program, the algorithm say that you can align A to A, A to A, and you insert a gap at the T here, and then you get C and G. This is the best you can do. You can't do better than this one. For example, this is the optimal, the best alignment. You can try to compete with this computer and then you see if you can get a much better result. But this one already gives you the best score, which is 2. So if you can come up with the better score, you'll win, okay? You can try that by yourself. But what happens if we actually add 2 more nucleotides to the sequence? Sorry, maybe 1. Okay, so this is what happens when I add 1 more nucleotide. You see, the alignment just add another gap here so that it's going to align with my T. And now the score is 0 because every time you add a gap, it's going to be deducted. The score will be deducted by 2, right? So the score is from 2 now it comes down to 0. So what happens if I add more? If it get longer and longer. Okay, you can see now this one is quite interesting. You can align this sequence. Sorry, this is not a good example. How about this one? Okay, so this is much better. And this one, you can see that you can actually get 2 alignments that have exactly the same score. So this is, the best the program can do for you. It gives you 2 alignments with the best score, and it cannot tell you which one is the best, but it gives you the best score, which is -1, but there are 2 ways that you can align to get the same score. So this is the problem, right? When you use program like this, it's not going to give you like the best one. It's just, it gives you the best score, but you don't know which one is the best anyway, because there are 2 ways that you can align the sequences. And this is only a few nucleotides. So it's going to get better if you have a longer nucleotide, right? Because if the nucleotide is longer, the possibility that you're going to get many alignments with the same score is going to be lower. So if you work with small or short nucleotide, you are more likely to get many alignments with the same score. But if your nucleotide is longer and longer, it's going to be more specific, and you're gonna get, most of the time you're gonna get maybe only 1 best alignment. So, but this is the best. This is the algorithm that finds you the best alignment. So it's work fine for short sequences, but not a long sequence. It's gonna take a while. And if you want to compare like 1 million sequences in the database, it has to do this for every million nucleotide sequences. It's going to take you a day. That's why BLAST doesn't use this approach directly. Actually come up with some other ways that can apply this approach with this algorithm with some other ways, so that can speed up the process. But at least you can see how it actually, what's going on, how we can tell the computer to align the sequence for us, even though the computer doesn't know anything about the DNA sequence, but it can align a sequence that gives you the best score. So this is what the computer scientists, like, they do all the time, right? They come up with the tricky way, the smart way to tell computer to do what they want. And if you're interested, you can read more about it. There's so many YouTube videos that are pretty easy to understand. Okay, let's get back to a little bit more theory before we go to the hands-on practice to end the day. The point number 2, okay. This is point number 2. There can be more than one alignment with the same score, and you just saw this one already, right? Many times you can get, sometimes you can get more than one alignment with the same score. So this is normal. It happens a lot. Okay, so let's do the pairwise alignment example. We're not going to use BLAST for now because we just want to compare 2 sequences, and we want to see how we get, what we get from doing global alignment and local alignment, so you can see the difference. So what I want you to do now is that you go to this website, go to this website again, copy and paste the URL and go to the website. You can try it so that you can, you know how to use the other tools. So this website is in EBI, right? So they have so many tools that you can use to align sequences. You can do both pairwise sequence alignment and multiple sequence alignment. And you see, this one is the first one in global alignment we call the needle. The needle is actually from the Needleman Wunsch algorithm. So you see the name and you know that they actually implement the Needleman Wunsch algorithm to this tool. And you can just click the needle to do the global alignment. And it's gonna ask you, like, this is the protein or DNA. You can just choose the protein, and then you just copy the sequence from the PowerPoint, slide number 39, the PK3CA bovine sequence. Just copy and paste. See, this is a protein sequence, so it's just the list of amino acid. And then copy another protein. It's called the protein kinase in bovine as well. So we're going to align these 2 proteins that have different lengths using the global alignment and see what happens. And then just hit submit. We're not going to change any default settings. Just going to use the default setting and then just submit. Yeah, it's going to run for maybe less than a minute. Okay. And you're going to get the alignment like this. And you can see, there are a lot of gaps, right? Because you do global alignment. The global alignment will align the sequence end to end. So the alignment length will be the same. That's why lots of gaps are inserted into the alignment. And you see these are all meaningless because, the sequence matched with the gap. And then this is the start of here is the start of the sequence, the second sequence. I start from here, and you can see some of them are aligned, like the one with the vertical line is perfectly matched. And the dot actually represent the alignment of the amino acid in the same group. So if you have 2 dots, it means that they are more likely to be substituted because they are similar in terms of property, physical chemical properties. So you can see most of them are actually aligned due to the chemical properties, but they're not really similar. And you don't see anything that, okay, just keep this one open, and then we're gonna try local alignment. You know, so just open another tab. I'm gonna open another tab and then go to the same website and choose the local alignment tool. Sorry. Go to the NCBI. And if you go to the local alignment tool, you see there are so many other tools, like so many tools that you can choose. Like the water is actually implemented the Smith-Waterman algorithm, which is invented 11 years after Needleman-Wunsch algorithm. But this one, I'm going to use the L line. The Lao line is actually finds internal duplication, blah, blah, blah. We're going to try to use this one for local alignment. And I'm going to just copy and paste the sequence, the same sequence again. And then the second sequence. Okay, just copy and paste 2 sequences like we've done before. And then we just submit. But this one is going to try to align locally, which means that it's going to look for a good alignment within the sequence. It doesn't care if the sequence align, like all other part of the sequence align or not, just the local part. So the result is going to be quite different from before. In the last tab, you can see that this one you have only one alignment, right? The best alignment that the program gives you. And you can see here, you know, the length of the alignment from the global alignment is 1,167. And that's only like 5.7% identity. So these 2 sequences only identical about 66 amino acids that identical, that align perfectly, perfect match. But they're similar 99.8%. The similarity means that those amino acids actually have the same properties. So they're similar, they're not identical, but they're similar. So a number of similar amino acids actually kind of more than identical amino acids. And this one gives you like 9.8%. And you actually inserted 915 gaps. That's a lot for global alignment. It's kind of not that useful. But when you look at the local alignment, this is going to be a little bit different because for the local alignment, you can have many regions that actually align well, but not the entire sequences. That's why you have many lines of the output, right? Many lines of the alignment output. Some of them have, depending on each region, or some of them have high similarity, some of them have low similarity, for example. And the length of the alignment is a lot shorter than the global alignment. Like this, the first one, only 49 sequences are aligned. The second one, only 43 are aligned, for example. And you can look at each, you know, alignment result. You can see that, you know, it only gives you the part that actually align, doesn't care about the rest. What I want you to know is that this part is actually very important, you know, here. And this one doesn't show up in global alignment at all. You can go and look at it. The position 920 and 930 are aligned to 170 and 180 in another sequence. Actually, this part actually important. It's more like a domain protein that highly important in the kinase protein, and it's actually found in many kinase protein. So if you look at many different kinase protein, you're going to see this part. And it's only picked up by the local alignment, not the global alignment. So this is just an extreme, extreme example to give you like what happened if you use the wrong kind of alignment. You're not gonna get the result. Okay. Okay, here, this one is the result from the local alignment. And actually, the one that I highlighted here is the functionally important residues and repeat found in nearly all kinase domains. So if you look at all the protein kinase, you should see, these, residues, because they are very important. But if you look at the global alignment, you're not going to see it. This is what happened when you do the global alignment. That this part of the sequence actually mapped to some gaps. Instead of the DRHN here, it actually mapped to some gap, because in global alignment, it tries to align the sequence end to end. So sometimes it's going to give you the wrong result. So that's why I said, if you care about the domain within the sequence, you should use the local alignment. Okay, the next one is going to be a tool. So I think we can continue this one tomorrow. And what we're going to do tomorrow is that we're going to use BLAST a lot, and then we're going to try to design a primer using the primer BLAST. And we're going to talk about some other tools that BLAST offer, because there's so many different kind of BLAST. BLAST is actually a set of programs. It's not a single program. It's a set of programs that you can use to do many different things. And we're going to talk about them tomorrow. And we're going to play with BLAST tomorrow, like some of the BLAST tool and some parameters that we can play with, and how to interpret the result. Okay, so we're going to talk about like all this stuff tomorrow. And actually BLAST kind of it gets updated a lot, so every year when I teach BLAST program, it has a new things, and it changed some rules, and it has some updates that I have to keep like I have to follow. I have to like update myself all the time, because it changed a lot. It's quite popular. It's quite popular. So many people are using it, and it kind of update a lot, and it changed all the rules a lot. So I would encourage you to read the manual if possible. Read the website a little bit about what actually happening, so you know what you can do with BLAST. Okay, so tomorrow one of you is going to present maybe for 15 minutes, okay, at the beginning of the class, and we may discuss about the paper a little bit, and then we're going to try to use BLAST and primer BLAST to design the primer. Designing the primer is not really what I do all the time, because I don't usually work on the wet lab, but it's something that you might get to use when you work on the experiment. You should at least know some the principle of using the primer BLAST to design the primer. But I can tell you that designing the primer is kind of it's quite specific to what you want to do. Like if you want to work on the virus, the gene or the genome of the virus, you have to know something about the virus in order to design the primer. So I don't think that's the rule of thumbs on how to design a good primer. I mean, there's some like principles, but when you work on specific organisms, specific gene, you're going to have to learn more about those sequences, like what is the characteristic of the sequences in order to design a good primer, because sometimes it doesn't work like you expect. In theory, it might work, but in practice, it doesn't work, for example. So designing primer is not really it's not very straightforward. You know, you have to know something about the sequences. You have to try, even though the program say that this is the best, it might not work at all, for example. So I may not be able to give you specific detail or instruction on how to design the primer in a specific way, but at least you know the principle and you can apply. Because I went to the bioinformatics research one time for the virologists. So basically people in Thailand who work on the viruses, and they have a very, very specific, you know, way to design the primer. You know, they look at so many different things that I don't think people who work on other fields will care. But because they know a lot about the virus, they know a lot about the genes of the virus and the genome of the virus. So different viruses will have different characteristics as well. So you have to kind of know a lot about biology of the organism or gene in order to design the primer well. Okay, see you next time tomorrow at 1 p.m. And don't forget about the assignment, okay? It's not a lot. Okay, see, I'm actually recording this video, so I'm gonna upload the video later, okay? And in case you want to review later on. Okay, see you tomorrow. Bye.